Hello, my name is Lars Bode, and I am a professor of pediatrics here at UC San Diego in California. And I'm going to talk today about human milk oligosaccharides with specific focus on immune system development. Before I get started, a couple of words about disclosures. Most of the research in my lab and our research center is funded by the NIH, NSF, or the Gates Foundation. And I'll start out with this sentence from the Lancet Breastfeeding Series 2016 that says that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, which comes along with economic savings of about $300 billion. And the question for us is, why is that? What is in human milk that makes it so powerful to save lives? And how does this work on a deep mechanistic level? So let's lift the curtain behind this statement here and see what is in human milk that makes it so powerful. And let's just focus on the macromolecules. There's protein, fat, lactose as the predominant milk sugar, and then there is oligosaccharides. 5 to 15 grams per liter, which often exceeds the total amount of protein in human milk. On the other hand, in bovine milk, there's around 100 to 1,000-fold less oligosaccharides, and they're very different in structure from human milk oligosaccharides as well. Since bovine milk is the basis for most infant formula, there's hardly any human milk oligosaccharides in infant formula, and that's a huge difference compared to breastfeeding. Now, what are these human milk oligosaccharides? A couple words on structure. Oligosaccharide means really a few sugars. So if we look at that, the table sugar that you know, the sugar that you have in your food, that is something that we really don't want to have in the infant space. The structure looks like that. It's a disaccharide. If we compare that with a human milk oligosaccharide, you see there is way more complex structure, and this is a fairly simple human milk oligosaccharide. To make that a little bit easier, we give each building block a different symbol here and then put them together in this shape and form. And there's really only 5 building blocks. There's glucose, galactose, N-acetylglucosamine, fucose, and sialic acid. And it's almost like Lego bricks. Depending on how I put these building blocks together, I either get a small car or a small plane, and we all agree that planes and cars do different things. And it's very similar with oligosaccharides as well. Structure of the oligosaccharides, the way I put these building blocks together determines their function. Now, without wanting to overwhelm you, showing you a little bit about the HMO blueprint here in the blue box, and then a few examples in the white space around it. All oligosaccharides start with lactose at the reducing end, and then they get extended, usually with disaccharide units, and that gives us either very long linear structures or branched complex molecules. Now, those structures can be fucosylated, so we add fucose in different linkages, either to lactose alone, and we get 2 and 3 fucosylactose, or to the growing chain and get different structural isomers. We can also add sialic acid, which introduces a negative charge, and we can add sialic acid either to lactose or to the growing chain and add more than 1 sialic acid. There's around 150, 200 different oligosaccharides that have been identified so far, and it's key to remember that every mom makes a different set of oligosaccharides. This is exemplified here in data from the Canadian child cohort, where we analyzed around 1,200 different milk samples on the x-axis here, and then different oligosaccharides in different concentrations in different colors on the y-axis. And you can see how different that is from subject to subject. And also the relative abundance of these oligosaccharides is very different from mom to mom. It's almost like a thumbprint. However, it's very different from subject to subject, but the concentrations are remarkably constant over a short period of time, here shown within a week, and on the right within a day or within a few hours in a day. So remarkably constant over a short period of time and very different between different subjects. Now, over a longer period of lactation, those concentrations change within the subject, but in a small time window, it's fairly constant. This is a different way to look at oligosaccharides, now represented in what we call principal component analysis, where every dot in this three-dimensional space represents the human milk oligosaccharide concentration or composition in a given milk sample. The closer those dots are together in this three-dimensional space, the more similar the HMO composition overall, and the further apart these dots, the more dissimilar the oligosaccharide concentration. These are samples represented from multiple different sites around the world, and actually the oligosaccharide composition is different in different parts of the world. We're trying to understand what drives this diversity of oligosaccharide composition between different individuals, how that impacts infant health and development, and whether we can learn from the maternal infant space and develop new therapeutics for adults or really for people of all ages. And for the sake of this presentation, I'm going to focus specifically on infant health and development, and specifically on the immune system. So how do these oligosaccharides work? First of all, we need to know what happens to the oligosaccharides once they're ingested. And really, oligosaccharides go in, they're very resistant to the low pH and to the pancreatic and brush border enzymes. About 1% is absorbed in the small intestine and appears in the urine, and the rest is degraded either in the colon by bacteria or is really excreted intact, and we find it in the diaper. Now, what do these oligosaccharides do? Here's a schematic that shows you the interactions between oligosaccharides on the top in the intestinal lumen, with good bacteria in green on the right, potential pathogenic bacteria on the right, and then the intestinal epithelial cells on the bottom, and then beneath that is, or underneath that is, the immune cells. So we know that oligosaccharides serve as prebiotics, so food for certain bacteria that may have a benefit to the host. But we strongly think that oligosaccharides are more than just food for bugs. We know that oligosaccharides keep certain pathogens in check by being direct antimicrobials, but also by being anti-adhesives, so protecting the host by preventing adhesion of certain pathogens to the intestinal epithelial cell surface. Oligosaccharides also have direct effects on epithelial cells, modulate their program, which then can change microbial communities in the gut as well. And we know that there is direct effects on immune cells, so being absorbed and interacting with receptors on immune cells directly, which also can have an effect on bacteria in the lumen. There can be local effects in the gut, but also systemic effects, because 1% of these oligosaccharides is absorbed and makes it into the systemic circulation and really flushes the entire system. And we believe that this is just the beginning. This is just the tip of the iceberg. We think there is so much more to discover when it comes to the effects of oligosaccharides. Now, how do we study that? We use everything from in silico models to ex vivo models, use animal models with all the restrictions that they do not really recapitulate human health. We use a lot of data on human cohort studies where we associate human milk oligosaccharide composition in mom or in her milk, and then infant health as outcomes. And last but not least, once we identify individual oligosaccharides and what kind of effects they might have, we then do clinical intervention studies to really confirm that these individual oligosaccharides have the given effects. And all that, of course, is not happening just in our lab. It's really happening with a network of collaborators from around the world. And instead of giving you like all kinds of examples at a broad overview, let's focus on one specific disease here for a minute to show you really how we study human milk oligosaccharide effects. And that is in the preterm space, and specifically on necrotizing enterocolitis. NEC is still about 5% incidence in all very low birth weight infants. It's an intestinal disorder that happens rapidly and destroys parts of the intestine. On the other hand, we know that human milk feeding protects the preterm infant from necrotizing enterocolitis, and infants that receive human milk are at a 6 to 10-fold lower risk to develop this devastating disease. Now the question is, what is in human milk that makes it so powerful, and is it potentially human milk oligosaccharides? And showing you just this in one slide here to develop how we studied that and how we figured out which oligosaccharide is responsible, we first isolated oligosaccharides from pooled human donor milk to get all oligosaccharides, the entire garden variety of oligosaccharides from different milks, pool that, isolate the oligosaccharides, and test oligosaccharide feeding in an in vivo model, in a rodent model of necrotizing enterocolitis. We then found that there is a huge effect on survival and reducing pathology scores, and then wanted to see, well, which oligosaccharide is it? Is it any of the 150 different oligosaccharides, or is it one specific one? So we did that by using multidimensional chromatography, separated the oligosaccharides first by charge, then by size, then by epitope, and identified that the responsible oligosaccharide for the effect that we see here is called LNT, short for disilolacto-N-tetraose. We further confirmed the chemical space around this structure. So for example, if you take off the terminal sialic acid, the compound does no longer work. But we often get the criticism saying like, well, this is a rodent model. This is not really clinical necrotizing enterocolitis the way we see it in our NICUs. So to address that criticism, we designed a mother milk infant cohort where we specifically recruited moms with babies that were preterm, under 1,500 gram, and then really followed them for the first 28 days of life, took milk samples about every second day, milk that was fed to those infants on those days, and then saw, okay, which infant is developing NEC, and are the oligosaccharides in the milk that was given to these infants any different than the oligosaccharides that were given to infants that did not develop NEC? And to make a very long story short, we found indeed that there is a match. So the oligosaccharide LNT that is protective against NEC in the animal model is at lower concentrations in the milk that was given to infants that later developed necrotizing enterocolitis. We've seen this in one cohort in North America. We've seen it in the meantime in a second cohort in South Africa, and we're about to publish a paper from a third cohort with about 35 different NEC cases or infants with NEC. And we see the very same result again, LNT being particularly low in those infants that develop NEC. So this gives us great confidence to move forward with clinical studies, but also allows us to study the mechanism in this animal model. So we have one specific oligosaccharide that we identified to be protective in this specific context in this disease. So we have one specific HMO that is associated with lower risk in preterm infants. The same oligosaccharide improves survival in an animal model, which is a great opportunity, of course, to develop new therapeutics around this oligosaccharide LNT, but also now gives us an opportunity to screen mom's own milk or donor milk products that have low levels of this protective oligosaccharide and say, well, maybe we're not feeding this specific donor milk batch. Let's take another one that has higher levels of LNT to make sure that our infants do not develop NEC. Now, that's just one example, and it's an example of many. So there's so many different studies that have been done either with us or with other labs, where we look specifically how oligosaccharides impact short and long-term infant health. And I'm giving you just a few examples here of what has been done. So for example, in the context of diarrheal disease, where we still lose about 2,200 infants under the age of 5 almost every single day. And we know that human milk is protective, but here now we show that individual oligosaccharides, here for example, 2-fucosyl-lactose, being protective against several pathogens, Campylobacter, enteropathogenic E. coli, and others. LNT, for example, was shown to be protective against group B streptococcus, but also against Entamoeba histolytica. And you see on the side here what symbols pop up. Some of this data is done either in tissue culture or in animal studies or in cohort studies. So you see there is some context where we have more data than in others. For some, we have the entire line from tissue culture through animal models and then going into cohort studies. Another example here is 3SL and 6SL in the context of rotavirus infections. On another topic, for growth, we see that 2-ethal is associated with growth in infancy and early childhood. And neo-LNT is also associated, but in the other direction. So there's positive and negative associations. And we've seen this, so the ratio between different oligosaccharides is important. And we've seen this in different cohorts now, in either smaller cohorts, pilot cohorts, but we can confirm that in larger studies where we have almost 1,000 mother-infant cohort pairs in these cohorts, where we see over and over again the same associations between 2-ethal and neo-LNT on one hand and infant growth on the other hand. 3-ethal and 6-ethal on the other hand were shown to be associated with malnutrition, so growth in when moms have malnutrition. Then another space is neurodevelopment and cognition. And again, 2-ethal stands out either in animal models or in cohort studies. 3-ethal and 6-ethal in other studies are protective or beneficial when it comes to memory and learning. But now, last but certainly not least, let's talk about the immune system and immune development. Lots of studies showing that, for example, defucosyl lactose is involved in cytokine release. Or we have shown quite extensively now that 3-sial lactose is somehow involved with chronic macrophage inflammation. We've seen before that specific oligosaccharides that have to carry both sialic acid as well as fucose can block leukocyte activation and extravasation, which might be important in an overly active immune system in the context of necrotizing enterocolitis. Other studies show that oligosaccharides are associated with lymphocyte maturation and a Th1, Th2 balance, or in animal models here showing that specific oligosaccharides stimulate the cytokine IL-10. So all this is from in silico or in vivo models, but now let's focus on a few human cohort studies that we have done together with many other investigators here. So here's a study that we did together with Kirsti, Javin, and Seppo, where we looked at cow milk allergy and whether mom's oligosaccharide profiles can somehow modulate the risk of cow milk allergy in infants. And looking at all these different oligosaccharides, really there's one that sticks out here, LNFP3, that is much, much lower in those infants that develop cow milk allergy. Then there's another study that we did together with a child cohort and Megan Azad's team, where we found that specific human milk oligosaccharide profiles are associated with food sensitization. So here it's important to remember or to show that we did not see any associations with individual oligosaccharides like we did in the previous study, but we found that oligosaccharide profiles, so how different oligosaccharides are linked together in different ratios to each other, really predicts, in this case, food sensitization. And a very similar result we found in a very large study here with a group at Melbourne, where we looked at oligosaccharide profiles and allergic disease all the way up to 18 years of life. So these milk samples were collected in the 1980s, and these infants are now in their 30s. And we had follow-up data here all the way up to 18 years. And again, we find that it's not one specific oligosaccharide that is associated with these different allergic diseases, but it's groups and clusters of oligosaccharides that can predict whether a person develops a certain allergy or not. If you want to read or hear more about this, there's a great review article here on the immunological effects of human milk oligosaccharides. First author being my colleague Vasilis Triantis, published in Frontiers. And there's another one that is specifically a systematic review on human milk oligosaccharides and associations with immune-mediated diseases and infection in childhood. So bringing this all together, what are oligosaccharides and what do they do and what do they have to do with the immune system? So again, HMOs are the third most abundant solid component of human milk after lactose and lipids, and often more abundant than human milk proteins. They are a group of complex sugars, glycans, and there's about 150 to 200 different ones. And it's important to remember that every oligosaccharide might have different functions and that every mom makes a different set of oligosaccharides. There is small oligosaccharides, so we can distinguish them between small and larger. So small being the ones that are just modified lactose. So that's your 2-FL, 2-fucosyllactose, 3-fucosyllactose, 3-siallactose, or 6-siallactose, and then the larger ones. So that's the LNTs, the neo-LNTs, and the derivatives of that and the longer structures. Then there can be neutral oligosaccharides and acidic oligosaccharides. So those that either do not carry sialic acid or carry sialic acid. And again, sialic acid introduces a negative charge, so it changes the structure quite a bit. Then they can be fucosylated or non-fucosylated, so carrying fucose or not carrying fucose, which again is a structural feature of the oligosaccharide and might change the function. And it's key to remember HMO structure determines function. Not all oligosaccharides do the same, are the same, and have the same effects. HMOs are not digested by the host, in this case, the baby. There is local effects within the gut, as well as systemic effects after absorption. 1% is absorbed and makes it all the way into the urine. 1% sounds little, but 15 grams per liter and getting oligosaccharides in every 2 to 3 hours during the feeding periods, that's a decent concentration in the systemic circulation. HMOs are prebiotics, so food for bacteria, but they're also anti-adhesives, antimicrobials. They have direct effects on epithelial cells, and they have direct effects on immune cells, either locally in the gut or systemically after absorption. HMOs can affect the outcomes like infectious diseases, diarrhea. They can affect growth. They can affect neurodevelopment and cognition. And they can affect directly immune system development and response. And we strongly believe that this is just the tip of the iceberg. We are now just trying to figure out what these oligosaccharides do on a molecular, on a cellular level, and then how they are related to infant health and development. And it's important to remember that sometimes we have specific individual oligosaccharides like 2-FL, like 3-SL, like 6-SL that have specific effects, and these effects are structure specific. So you cannot exchange one oligosaccharide for another. These effects are highly structure specific and follow a dose response. So the higher the oligosaccharide amount, the better the effect. And these effects are likely receptor mediated. So there is a specific receptor either on the host or the microbe, and the oligosaccharide, this one specific oligosaccharide, fits in or doesn't fit in, but if it fits, then it has an effect. So highly structure specific. And other times, and we've seen this quite often now in the space around allergy, asthma, wheezing, where we need mixtures of different oligosaccharides, so composition profiles to be effective. So the relative abundance, the ratio of different oligosaccharides to each other, that's what matters. And the mechanism is likely indirect through shaping microbial communities where you can imagine that one microbe prefers this oligosaccharide, another microbe prefers that oligosaccharide, and the community as a whole feeds really on a mixture of these oligosaccharides, and then we have a beneficial effect. Or they shape immune system responses where different immune cells, different immune receptors are involved. Different oligosaccharides trigger these receptors in an orchestrate almost, and then the effect follows along. So in any case, we need a combination of preclinical, cohort, and clinical trials to fully assess and validate the effects and functions of oligosaccharides, either individual oligosaccharides or mixtures. And I'll leave you with that. And thank you very much for listening.